Hi, I'm Richard Weinberg from the USC School of Cinematic Arts, and I'd like to talk to you about Project Tele-Microscopy for teaching biology. Well, every scientist, biologist, and healthcare worker knows that the microworld is critical to all life on the planet, but high school students can't understand it, they don't appreciate it, they can't see it, and they don't have university faculty to help them understand it. So we've developed this project to deal with that. Here's where I work, USC School of Cinematic Arts. And what I've been doing is integrating technology that was developed for the entertainment industry, taking a digital cinema camera, integrating it with a very high quality microscope, and connecting it to the network so that we can stream ultra high resolution images from this system to remote places like a STEM school in Chattanooga, Tennessee, where we just started a new biology class. So to get there, we're using a variety of networks going from USC through Scenic in California, through NSF networks, through Internet 2, the AL2S, to Genie Rack in Chattanooga, Tennessee, and with help of US Ignite and the National Science Foundation, we've been able to transmit these images across the network. So here's the basic system. We have this research microscope built by Olympus, and I'm integrating digital cinema cameras, sometimes from Red Digital Cinema, sometimes from AJA Scion, capturing them into the computer, and we can see them locally and transmit them to a remote site. I'm also wrapping this whole thing in essentially a television production studio so that we can do additional video processing and eventually take the microbiology professor and put him inside the microscopic slide. So we're working on that. Then we're also coupling that with a video conferencing system so that we can have a two-way dialogue between marine biologists at USC and the remote students. At the remote site, we invert the process, pick up the video, and run it through a program called UltraGrid to create essentially a copy of what we're seeing just a fraction of a second later, and there's only about 30 milliseconds delay from USC in Los Angeles to Chattanooga, and we have a simultaneous video conference going so we can interact remotely. So here are a couple of setups at USC, and in fact today we have the Olympus microscope set up here with the Scion camera. So we're utilizing this technology developed for the entertainment industry for a new type of education. Here's one of Dr. Karen's graduate students, Paige, operating the microscope in Los Angeles. But we've also set up the ability for the students in Chattanooga to operate the microscope remotely, which really helps engage them in the process. They can move the stage, to move the image, they can focus the microscope, change the illumination, and change the objectives on the fly. And they do that using this program from Olympus called CellSense, which kind of is paired with the microscope. And it's what we would use in Los Angeles at the local station. And using a program from HP, their RGS, we were able to remotely operate that. So just last week we had the first class with students in Chattanooga. You see them here, and Dave, Karen, and Paige here in Los Angeles. And we found the students were really engaged in this process. They had never seen the kind of microscopic images in actual life, moving around, interacting with one another, seeing who's eating whom, how they move. So it's really an exciting experience for them, and being able to control the microscope remotely really helped to engage them in the process. They really liked that. And it took all of about a minute to show each student how to use it, so it was a very fast process. And some more happy students in Chattanooga. And how this all came about, I've been working in this area for almost 10 years since the Red 1 camera came about. I was a microscope hobbyist, and when the digital cinema era started, I thought, well, it'd be interesting to see if I could integrate a digital cinema camera with a microscope, and sure enough I could, and I've been working in this area ever since, using better and better microscopes And then I started broadcasting these images over the network because I'm involved with a group called CineGrid that does high-speed projects using the optical network system around the country and around the world to use another microscope here to broadcast from USC and Los Angeles to other schools. And here's another camera, which is a 4K camera that can do 4K 60 frames per second. And with this, we were able to stream from USC to Tokyo, actually winning a prize from CNEC. And here are some of the microscopic characters that we've collected. So Dave Karen at USC is a marine biologist, and his group brings back plankton toes from the Pacific Ocean. And so I've been using those as well as microscopic samples from pond water to create a whole series of movies in the pond, microorganisms, and the dramatic sequel, Microorganisms 2. And one of the things that really surprised me and made me happy was how young children are really drawn to this. So we had an event in Los Angeles, the enormous microscopic evening, and I set up my stuff, and even little children were just drawn to these microscopic images. Really fascinating. In fact, people of all ages are really drawn to them. And I've been displaying these on large video walls like this one at UCSD, Cal IT2. And at the opening of the new Exploratorium building in San Francisco, they got in touch with me to do a projection onto the building. And for this, a company in Canada manufactured a microscope slide where the facade of the building was engraved into it. I put the microorganisms into that, and they had to swim around the channels. So when you project it onto the building, the microorganisms are actually interacting with the windows and the doors. And we had a 2-night presentation of the grand opening of the new building in San Francisco, and it was quite amazing. And I've also found that working with microbiologists who are getting plankton samples from the Pacific Ocean, if you leave that water on a microscope slide overnight, it'll crystallize into these beautiful forms. And that got me interested in growing my own crystals and filming them under time lapse. And here you can see a few stills from those. And with the microscope, we can also look way back in time. So here we're looking at sections of meteorites that predate the Earth. So they're 4 billion years old or more. And I'd like to thank all the wonderful organizations that have made this possible. And thank the National Science Foundation for the digital telemicroscopy in support of teaching biology grant, in addition to the other NSF grants at USC that have made this work possible. And now I'd like to switch back from this to USC, where we have a sample under the microscope at USC. It's just like the one that you see here, but it's on the system remotely. And so we're able to stream live high definition images here while we were simultaneously recording 4K images in Los Angeles, which we can then use to create movies. We can send those to the students to edit and further learn about what they saw during the class. Here you're seeing a rotifer that is on a microscope slide that is from some pond scum in Los Angeles. And what we're able to do with this screen now is to control the microscope remotely. So if I want to move the sample here, if I just click on this little widget, I can reposition the sample. I could change the focus, or I could change the magnification. And this allows us to have an interactive experience with the students. So they see something they're interested in, they can steer themselves right over to it, they can ask questions, and we'll also be doing research where students from Tennessee, from Chattanooga, will take samples from the Tennessee River, which their school is overlooking, And they'll be running experiments. They'll be looking at what's happening in the water there under their own microscopes. But they'll also be sending us overnight delivery samples of what the water is like, what happens when they try different experiments with it. Then we'll be able to put it under our microscope. And our marine biologists at USC will be able to take a look and tell them, okay, well, now you've got this kind of organism here, this kind of organism here. And because you put in these type of chemicals, pollutants, or whatever, it's caused this change in the ecology of the sample. So, we normally have no access to the Tennessee River, and students in Chattanooga have no access to the Pacific Ocean. So it'll be a good exchange of information. And my hope is that this system will encourage students to be more interested in the sciences. I think a system like this, because it includes so many different types of technologies, microscopy, computers, networks, optics, as well as tying into the sciences like biology, material science, and a whole realm of scientific fields that the microscope is applicable to. My hope is that this will get students interested in science, technology, engineering, the arts, and mathematics. Thank you.